Welcome back to the deep dive. We're here again to, well, take complex stuff and really boil it down, find those essential bits, the surprising things, and honestly, the just plain fascinating knowledge nuggets.
hmm
Today, we are starting an incredible journey. I mean,
hmm
think about it, something smaller than a pinhead
Yeah,
that holds the entire plan for, a giant tree or a beautiful flower. The very, very beginning of a plant's life.
it really is.
We're diving straight into plant biology today, looking at how just one fertilized cell starts this whole intricate developmental dance
Yeah.
to create a living, breathing plant.
It's a story that's just full of precision and survival tactics, adaptation. Really one of nature's most sophisticated designs, I think.
Yeah.
So our mission today really is to trace this whole process,
Ah.
starting with embryogenesis. That's where the plant's basic body plan gets established, then moving through seed maturation and, crucially, dormancy.
Right.
And finally, how that new seedling actually emerges during germination.
And we're going to uncover some, frankly, amazing biological engineering along the way. Like, how does a plant just know which way is up for shoots and down for roots?
Here.
That's polarity. We'll look at the different stages the embryo goes through, what makes the seed just sit there, sometimes for decades or suddenly wake up,
Mm-hmm.
and even how studying these tiny genetic hiccups, mutations, has actually unlocked some really deep secrets about how plants develop. So let's dig in.
Let's do it.
Okay, let's unpack this. The very, very first steps. How does this whole amazing process even kick off?
Right. So it all begins with embryogenesis. That's the first phase of developing the seed. And what's really striking is how from that single cell, the plant's entire body plan is basically pre-programmed.
Wow.
We're talking two fundamental patterns being laid down almost immediately. First, there's the apical-basal pattern.
Okay.
Think of that as the main axis setting up where the shoot tip will be at one end and the root tip at the other.
Top and bottom.
Exactly, top and bottom. And second, you have the radial pattern. This establishes tissue systems arranged in concentric circles, kind of like layers in an onion, setting up distinct rings of tissues.
Okay.
And doing this groundwork early is why seeds are such a huge evolutionary win. They package this complete tiny plant, ready to survive tough times and get dispersed.
It's just amazing that the whole destiny, roots here, shoots there, is decided by one single cell division that's not even symmetrical. How does that first blueprint get drawn?
Well, that's where the zygote's very first division is absolutely crucial. It's not just any split, it's asymmetrical, and it's transverse crosswise. And that immediately establishes that vital apical-basal polarity.
Okay.
The smaller cell, that's the upper one, the apical cell, well, that's going to develop into most of the actual embryo, including the future shoot.
Right.
The larger lower cell, the basal cell, it produces this stalk-like thing called the suspensor. Think of it like a little umbilical cord. It anchors the embryo and helps feed it early on. So that very early commitment to top and bottom is just fundamental for everything else that happens.
So, okay, we have this initial split, setting the basic direction. Then the embryo starts to take shape first as a proembryo. Then it becomes this almost spherical embryo proper. What's going on inside that tiny ball?
Right, so now those concentric radial patterns really start to form. The embryo proper, even though it starts out looking pretty undifferentiated, it quickly sets up its core building blocks.
Yeah.
The three primary meristems.
Meristems. Okay. Those are like the stem cells, right?
Exactly like stem cells, yes. The outermost cells divide in a specific way to form the protoderm. That's destined to become the epidermis, the plant's skin.
Got it.
Then inside that, you get the ground meristem, which will form the ground tissue that's the bulk of the internal structure. And right in the very center, the procambium emerges. That's the precursor to the vascular tissues.
The plumbing.
The plumbing, exactly. Xylem and phloem carrying water and food. And these primary meristems, they run throughout the whole embryo, ready to differentiate into all the other tissues later on.
It's incredible. These tiny structures are basically pre-programming their entire future house plan.
Yeah.
And then we can actually see them take on different shapes as they grow, right? They don't stay spherical.
No, they don't. The embryo goes through these really distinct morphological stages. For Eudicots, those are plants with two seed leaves, like beans or sunflowers.
Right.
It starts as that globular stage, just a sphere, but then it develops into this unmistakable heart shape. That's when the two cotyledons, the embryonic leaves, start emerging. It really looks like a tiny green heart inside the seed.
Cool.
Then that elongates into the torpedo stage, where the cotyledons and the main axis stretch out. Now, in monocots like grasses or onions, you see a similar globular stage,
Hmm.
but they only form one cotyledon, so they tend to become more cylindrical as they mature.
Okay.
And importantly, all through these stages, those vital apical meristems are forming at the tips, the future shoot tip and root tip. These are the engines for all future growth of the adult plant.
And while all this complex shaping is going on, there's that helper structure we mentioned, the suspensor. It sounds simple, just an anchor, but you said it's more complicated.
Oh, much more. In flowering plants, angiosperms, the suspensor is definitely not passive. It's metabolically very active. It's a lifeline, really.
Yeah.
It actively pumps nutrients and also important growth regulators, like gibberellins, into that developing embryo proper.
Wow.
But it is short-lived. Usually by the torpedo stage, it undergoes programmed cell death, apoptosis. That's why you don't find a suspensor in a mature seed, though maybe remnants of that basal cell.
Interesting.
And what's really fascinating is what we've learned from mutants, particularly in the model plant Arabidopsis.
Ah, the lab rat of the plant world.
Pretty much. In mutants like twin, which stands for twin, if the main embryo's development gets messed up, the suspensor cells can actually start dividing again and even form secondary embryos.
No way.
Yeah.
Like actual twins.
Like actual twins or even triplets sometimes. It strongly suggests that the main embryo normally sends out signals to stop the suspensor from doing that, to keep it in its support role.
So this whole intricate development, it isn't random at all. It's controlled meticulously. That must point to a really deep genetic program, right?
Absolutely. I mean, our understanding of plant development has just exploded thanks to studying these Arabidopsis mutants. They let us pinpoint the genes involved. Scientists estimate something like 750 distinct genes are needed just to coordinate embryo development in Arabidopsis.
The 750 genes.
Yeah.
Just for the embryo.
Just for the embryo. And mutations in some of these genes mess up those initial apical-basal patterns. They cause specific missing parts. For instance, the gurke mutant might lack its shoot and cotyledons entirely.
Gurke, like cucumber.
Yeah, apparently it looked a bit like one. Then the monopterous mutant might be missing its root. Others affect the radial pattern, maybe preventing the epidermis from forming properly. And still others are needed just for the cell shape changes that allow the embryo to elongate. It's this beautifully complex, genetically orchestrated dance.
Okay, so we've walked through the blueprinting, the early construction. What does this little fully formed embryo look like when it's all packed up in the seed, ready for the next big step?
Right, so by the time it's mature, the embryo is pretty well organized. It's got a central axis, and attached to that are the cotyledons, either one for monocots or two for eudicots.
The seed leaves.
The seed leaves, exactly. And crucially, it has those apical meristems at both the shoot end and the root end, primed and ready for growth later. Above where the cotyledons attach, you've got the epicotyl. That's the embryonic stem part, and it might include tiny young leaves and the shoot apical meristem. Together, that whole embryonic shoot package is often called the plumule.
The first little bud.
The first bud, yeah. Below the cotyledons is the hypocotyl, another stem-like bit. And right at the bottom of that is the radical, the embryonic root, all set to grow downwards and anchor the plant.
Okay, and a plant needs fuel to get going. Where's the energy stored? Because you mentioned it's packed away differently depending on the plant type.
Exactly right. The food storage strategy varies a lot. Lots of Eudicots think of things like garden beans, peas, walnuts. They absorb most of the food storing tissue, the endosperm or sometimes perisperm, while they're still developing. So in those cases, their own cotyledons get really large and fleshy, and they become the main storage organs packed with starch or protein or oils.
So the cotyledons are the pantry.
They're the pantry in that case. But then you have other Eudicots like castor bean, where the cotyledons stay quite thin, almost papery. In these plants, the food remains stored outside the embryo, in the endosperm. The thin cotyledons then just act like straws, absorbing that food during germination.
Different strategy.
Different strategy. And monocots, with their single cotyledon, also vary. In grasses like maize or wheat, that one cotyledon is massive and highly specialized. It's called the scutellum. It's basically embedded right against the endosperm, perfectly designed just to absorb nutrients from it efficiently.
And every good packed lunch needs a container, right?
Yeah.
The seed needs its protective armor.
Absolutely essential. The outermost layer is the tough seed coat. It develops from the layers of the ovule, the integuments. Its main job, protection. It can be thin like paper or incredibly hard and totally impermeable to water.
Wow.
In grasses, things get a bit more complex. Often the outer covering isn't just the seed coat, but the pericarp, that's the mature ovary wall, fused together with what's left of the seed coat.
Okay.
If you look closely at a seed, you might see a tiny pore, the micropyle, that's often near a little scar called the hilum, which is where the seed was attached to its stalk inside the fruit.
I think I've seen that on beans.
Yeah, exactly. And monocots, especially grasses, often have extra layers of protection inside the seed coat for the delicate growing points. There's the coleorhiza, a sheath covering the embryonic root, the radical, and the coleoptile, another sheath protecting the embryonic shoot, the plumule.
Extra wrapping. Okay, so the embryo's fully formed. It's got its food, it's snug in its protective shell. But before it bursts into action, there's this really vital prep period, almost like going into suspended animation. What's happening during seed maturation?
Right. This is the critical third phase right after embryogenesis finishes. It's all about preparing for survival, potentially for a long time. First, there's a huge buildup of food reserves.
Mhm.
Starch, proteins, oils, they just accumulate massively in the endosperm or the cotyledons,
Stocking up.
totally stocking up. Second, and this is really crucial, the seed undergoes desiccation. It loses a massive amount of water, often 90% or even more, just dries right out.
90%.
Yeah, it's extreme dehydration. And finally, that seed coat hardens up, making this durable, sometimes almost impenetrable casing around the embryo and its food.
So what's the result of all that?
The result is the seed's metabolism just plummets. It slows right down, almost to a standstill. This allows the embryo to stay viable, stay alive for potentially very long periods. It might be just quiescent, meaning resting, waiting for water, or it might enter true dormancy. Either way, it's ready to wait for the perfect moment.
Okay, speaking of waiting, what does a seed actually need to wake up from that rest or dormancy and start growing? What triggers germination?
That brings us to germination itself the moment the embryo resumes growth. And it depends on a few key external factors. Number one, absolutely essential, is water. The process is called imbibition.
Imbibition.
Yeah, the seed literally drinks up water and swells. This activates enzymes that were dormant and triggers the making of new enzymes to start digesting all those stored food reserves.
Makes sense. Need water to get the chemistry going?
Exactly. Then you need oxygen. Some very early energy release might be anaerobic, without oxygen, but for the rapid growth of a seedling, you absolutely need aerobic respiration. That's why really waterlogged soil can actually prevent germination. No oxygen can get in.
Right.
Temperature is also key, and it's very species specific. Every plant has its preferred temperature range for germination. You know, a minimum, a maximum, and an optimum, often somewhere around 25-30 degrees Celsius for many common plants.
Okay.
And for some seeds, especially small ones like lettuce or many weeds, light itself can be the trigger they need to germinate.
But sometimes you give a seed perfect water, perfect oxygen, perfect temperature, maybe even light,
Yeah.
and it just sits there. Nothing happens.
I-
Dormancy, right?
yeah.
And it sounds like an amazing survival tool.
It is an incredible strategy. Dormancy is specifically when a seed actively fails to germinate, even when all those external conditions seem perfect. It's different from just being quiescent, where adding water is enough.
So it's like an internal off switch.
Exactly, an internal break. And the mechanisms are really clever, all geared towards survival. You can have coat-imposed dormancy. Maybe the seed coat is just physically impermeable to water or oxygen, like
Raincoat.
a tiny, tough raincoat. Or maybe it's just mechanically too rigid, the little root can't physically break through it. Sometimes the coat contains growth inhibitors or prevents inhibitors inside the seed from leaching out.
Huh.
Then there's embryo dormancy. This is more about the embryo's internal state. Often it comes down to the balance of plant hormones, especially abscisic acid, or ABA, which promotes dormancy, versus gibberellic acid, GA, which promotes germination.
Hormonal control.
Precisely. And a really common type of embryo dormancy requires after ripening. Some seeds, especially from temperate climates like, say, roses, apples, cherries, lots of woody plants, they need a period of cold, damp conditions, like surviving a winter. This triggers complex biochemical changes inside the seed. Only after that cold treatment can they germinate. It's a fantastic adaptation. It stops them sprouting in the fall, only to be killed by frost. They wait until spring.
That makes so much sense. So this isn't just a pause. It's like a calculated decision based on the environment. What does this mean for the plant's overall life strategy?
Well, if you connect it to the bigger ecological picture, dormancy provides immense survival value. It prevents that premature germination, making sure the offspring only sprout when conditions are genuinely favorable for seedling survival.
Maximizes the odds.
Totally. It also really helps with dispersal. Some seeds actually need to pass through an animal's gut. The digestive process breaks down the coat or inhibitors, breaking dormancy.
Wow.
And think about other environmental triggers. Some desert plant seeds will only germinate after a really heavy rain washes inhibitors out of their coats, ensuring there's enough water to sustain growth. Others might need mechanical scarification, like being tumbled around in a rocky stream cracks the coat. And then there are fire-adapted species, like manzanita in the California chaparral. Their seeds often need the heat of a fire to break dormancy, or the fire clears competitors, or even releases seeds from cones.
Amazing adaptations.
Yeah. Even just a gap opening up in a forest canopy can be a trigger. Seeds of plants that can't tolerate shade might lie dormant for years, waiting for enough light to hit the forest floor before they germinate. The whole strategy is just intricately tied to the plant's specific home, its ecological niche.
So once the signal is finally given the right conditions are met, dormancy is broken. Germination starts. What's the very first thing we usually see emerging from that seed coat?
For the vast majority of seeds, the first thing to push out is the root, specifically the primary root, which will often become the taproot. Its first job is absolutely critical. Anchor that brand new seedling firmly in the soil and immediately start taking up water and nutrients.
Anchor down, drink up.
Exactly. Now, in monocots, it's often a bit different. That primary root might be pretty short-lived. The main adult root system actually develops later from roots that sprout directly from the base of the stem, what we call adventitious roots.
Okay, so the root gets established, but then how does the delicate shoot, the plumule, make its way up through the soil without getting all scraped up or broken?
Well, the shoot emergence strategies are also really neat, and they fall into two main categories. First is epigous germination. Epi means above, geo means earth. So the cotyledons are carried above the ground.
Okay.
Think of a garden bean sprouting. The hypocotyl, the part of the stem below the cotyledons, elongates first, but it forms this distinctive hook shape.
I've seen that hook.
Yeah, that hook acts like a plow, pushing through the soil and protecting the really delicate shoot tip, the plumule, and the cotyledons tucked behind it. Once it breaks the surface into the light, the hook straightens up. Those cotyledons often turn green and photosynthesize for a little while.
Bonus energy.
Bonus energy, exactly, before they eventually wither away once the true leaves take over. Onions, which are monocots, do something similar. The single tubular cotyledon elongates and forms a hook, pulling the plumule up.
Right, and the other type.
The other type is hypogegous germination. Hypo means below, so the cotyledons remain underground. A classic example is a pea.
Okay.
In peas, it's the epicotyl, the stem segment above the cotyledons, that elongates and forms the protective hook. So it only pulls the plumule, the shoot tip, and first leaves above the ground. The cotyledons stay buried in the soil, serving as a food reserve until they decompose.
Less risk for the cotyledons that way.
Less risk, yeah.
Yeah.
And maize, corn, which is a highly differentiated monocot, has its own elaborate system. First, the coleohyza, that sheath around the root, emerges. Then the radical pushes through it. Then the coleoptile, the sheath around the shoot, is pushed upwards not by the hypocotyl or epicotyl, but by the elongation of the mesocotyl, which is technically the first internode of the stem.
Wow, complex.
It is. And the actual plumule leaves then emerge through a little pore at the tip of that coleoptile. Maize also forms extra roots early on, called seminal roots, from near the cotyledon, giving it more stability right away.
It really is this complex, perfectly timed escape from the soil, and once the seedling is successfully out and established, the growth just keeps going from those meristems, right?
That's right. Once it's established, that shoot apical meristem just keeps doing its job, producing an orderly sequence of new leaves, nodes where the leaves attach, and internodes, the stem segments between them. And then you get additional buds, axillary buds, forming in the axils where the leaves meet the stem. Those have the potential to grow out into branches.
Building the plant structure.
Building the plant. But it's worth remembering that this whole period, from the moment germination starts until the seedling is really established and photosynthesizing strongly on its own, that is the most dangerous, most vulnerable time in the entire plant's life cycle. They're incredibly susceptible to pests, fungal attacks, drying out. Just getting started is tough.
What an absolutely incredible journey we've followed today. I mean, starting from just one single cell, going through all those intricate stages of building the embryo, the strategic genius of seed maturation and dormancy, and then that final courageous push of germination to establish a new plant. It really is a profound testament to nature's design, its resilience, all happening on this tiny scale.
It really is. And, thinking about all this raises a pretty important question, I think. We've seen just how finely tuned every single stage is, from establishing polarity right at the beginning to the complex triggers needed for dormancy and germination. It's all adapted for survival in very specific environmental conditions.
Right.
So, how deeply intertwined are these processes with the actual habitat a plant lives in? And maybe more urgently, how might human activities, or especially the rapid pace of climate change, start impacting these delicate balances, particularly for those seeds that rely on very specific environmental cues, like a certain length of cold period, or fire or rainfall patterns cues that might just not happen reliably anymore in a changing world? What does that mean for their future?
That's definitely something to think about. A crucial connection between these fundamental processes and the wider world. Well, thank you for joining us on this deep dive into the very beginnings of plant life. We really hope you feel a bit more connected to these amazing, intricate life cycles happening quietly all around us every single day.